Then what is exactly happening in the biosynthesis of your citric acid? So mainly it requires the glucose source. This glucose will undergo glycolysis process. Then it will be converted into pyruvate component. So you have studied in TCA cycle already. So and in that you will know what is the exact structure. So the glucose will be converted into pyruvate, and this pyruvate will be undergo a further cyclic process which is called TCA cycle. We know that it will be exactly converted in either the oxaloacetate or malate. Then it will be completely converted into fumarate, and on further conversion, the possibilities of oxalate to the citric acid will happen. This is the exact TCA cycle along with your glycolysis pathway. So in glycolysis pathway, the glucose will be converted into pyruvate. This pyruvate, the possibilities are there. Pyruvate will be on releasing CO2, and also on the because of the enzyme pyruvate dehydrogenase, it will be converted into acetyl CoA. This acetyl CoA on further reaction, it will be converted into citric acid. So from this, the citric acid will be released from the cytoplasm to the medium. So this is the exact biosynthesis of your citric acid. So how we can improve the yield? Because in laboratory scale, we can say it is very quite normal. But in industrial purposes, we require some of the main sources which can able to convert that mm, uh, nutrient into a desired product. So for that, the substrate is mainly here only the glucose or sucrose. Only the carbon sources plays an important role in citric acid production. So some of the trace metals as well as some of the as an enhancer, then nitrogen sources have been used, but main source is the carbon source. The yield, they on studying all these things in laboratory manner itself, they found out that the glucose sucrose can be, if it is in 100 gram, the possibilities of obtaining citric acid will be 112 grams either in crystal form or else in the powdered form. So this will be definitely the sugar will be completely oxidized. So it requires an it is an aerobic process. Okay, so it will convert that into CO2. So these are the main parameters we should follow. The sugar concentration should be 10 to 25%. Magnesium, zinc, iron as the traces element, which is very, very important for this, and pH and dissolved oxygen. Then some of the ammonium salts, it is a nitrogen source, and also the time required is 150 to 250 hours. So, what are the possible carbon sources? We should know that on selecting this raw material, it should be completely because if the company is giving a very high priced product, it will be based upon only this raw material. Because this, if the raw material they are purchasing is very costly, then the product will also be very, very costly. So they will check from where they can get the exact low cost or cheap material, and also in a good quality to obtain a very good yield of our final product. So in this as a carbon source, they have used molasses, starch, then date syrup or cotton waste, banana extract, or else sweet potato pulp, then brewery wastes, and also the pineapple waste. So from all these, they will choose exactly for which microorganism we require the exact carbon source, and then they will go for the production of citric acid. Okay. So most of the commonly used are uh, molasses, cane molasses, and also beet molasses. So you know that the molasses, the name molasses means, you know that in sugarcane industry, while processing or extracting the sugar from the sugarcane, they will, after extracting the sugar, they will have one wastage, no, that will be, you can see also in shops, sugarcane juice shops and all, some of the waste will be come out of the extractor. So that is called as molasses. that will be used as a carbon source. Okay. So mainly they require the sugar concentration should be 12 to 25 percentage should be there. And if they are increasing the sugar concentration, then the citric acid production is very, very good. So from this study only, they confirm it in the laboratory scale, and then they went for this industrial purpose and all. Okay. So you can see in this diagram, the citric acid production will be there on the increasing the concentration of your sucrose. So on increasing the sucrose concentration from 5 to 15 and 20, you can see there is a slight variation, and also increase in the production of the citric acid will occur. So from this conclusion, they went for increasing the concentration level of the carbon sources, and then they went for the production. Next one is trace metals. So even though the carbon sources is very important, some of the trace elements will enhance the growth of your Aspergillus niger. So either the iron, copper, zinc, magnesium, cobalt, these are the main trace elements. But from this, they exactly required magnesium, iron, and also the zinc component. They have studied that only these three metal ions are very, very effective for the obtaining an yield of citric acid. Okay. So these ion, what is exactly these ions role is that it will promote the glycolysis process. So this pathway have been promoted, and also it will reduce the respiration process. Only they will concentrate on the conversion of the sugar into your desired product. Then we will move on to the pH. So the pH will be, we can say like it requires all the production requires. Next slide, pH slide. So all the production of either citric acid or lactic acid for anything, the optimum conditions should be maintained. This optimum condition will be based upon the pH, temperature, and also a dissolved oxygen present in that nutrient. Only based upon that we will conclude it, and also we can get exactly the concentration of our particular product. Okay. So the pH should be, if the pH should be above or below, it should be within a range 2.5 to 4. If it is below 2.4, the production of another component will occur. So either oxalic acid or gluconic acid will be happen. So only if it is at 2.5, the suppression of these two side products can be obtained, and exactly the citric acid will be produced. Okay. And if it is below what is above 4, then the accumulation of gluconic acid will occur. If the pH is beyond 6, then oxalic acid will get started producing it. So the pH range is very, very important. So it should be 2.5, and also it should not be beyond 4. Based upon the microorganism, we have to fix it either 2.5 or 3, or else 3.5 or 4, but not beyond the 4 range. Okay. Then dissolved oxygen. Definitely, this organism required, Aspergillus niger required oxygen supply for the production of citric acid. So this citric acid, that fermentation process should have an aerator or else sparger. It should supply the oxygen level. Only the oxygen should be supplied to it. So if there is any deficiency in oxygen, the production of citric acid will also occur. So dissolved oxygen plays an important role in the production of citric acid. Okay. Next one is nitrogen source. So there are many nitrogen sources. One is called ammonium salts, nitrates, and urea. These are the commonly used nitrogen sources for the citric acid production. Um, And we know that the carbon sources plays an important role, but this ion, ammonium ion or nitrate or urea, will also stimulates the citric acid production. That's why we are adding in the minimum amount of nitrogen source when compared to any other production. Okay. Then, what are the possibilities of production processes? First one is surface process, or another one is submerged process. So we know that surface process is nothing but mm, it will be like it will depends upon either the solids or the liquid. It will hang on to the solid surface or to the liquid surface and while start growing into it. But in submerged is that it will be present along with your media, liquid media, and it will be suspended in all the microorganism will not require any surface to hold on. So it will be in a solution itself. So that's why in most of the stirrer tank, then airlift, bubble reactor, bubble column reactor, or else packed bed reactor, we had only these type of